Welcome to another episode of Interview with a Biologist. I'm your host, Maria Locedo, and I'm joined today by Dr. James Bever, who is a KU Foundation Distinguished Professor of Ecology and Evolutionary Biology and a senior scientist at the Kansas Biological Survey and Center for Ecological Research. How has your week been so far?
It's been wonderful. Yeah, thank you.
Good. Yeah, it's 2 days into daylight savings time, so personally, I am struggling.
I can relate to that. Yeah. It's a rough adjustment.
Every year, one of these, it'll eventually go away, and it'll just be beautiful.
Yeah.
So, could you tell us a little bit about your research interests?
Yeah, I'd love to. So I study plants and all the microbes that they interact with. And plant roots grow down into the soil, and they're interacting with thousands of different kinds of microbes simultaneously. And some of them help plants grow, and some of them eat the plant and have strong negative effect. And how do we understand that net effect and the dynamics of that system, and how plants try to balance all of the beneficial and the non-beneficial interactions? That's sort of what we study.
Along with all of your research work, I believe you also teach some courses at KU. Can you tell us a little bit about those courses?
Yeah, I teach lab and field ecology, and I teach a upper-level course on concepts in ecology. They're a lot of fun.
Yeah, and I believe the field ecology class, does that mostly take place at the KU field station or simply around Lawrence?
It's the KU field station. We use that a lot. We use other resources around Lawrence as well. It's traveling, taking students around in a van and doing exercises with that.
That sounds amazing. So was there anything, like as you got it started, was there something that really drove your research in like that interest in soil ecology?
Yeah, good. That's a good question. So I was starting out trying to think about how plants, whether plants from a local environment would do better in their local environment, maybe because they reinforce how the beneficial microbes, and or would they do worse because the microbes would overwhelm them? And that simple question, which is something that all gardeners might relate to, actually developed into a whole avenue of research where I evaluate the net effect of how microbes impact plant growth and how that relates to the structure of plant communities and the productivity of plant communities. And it's been a lot of fun, and it's been a sort of an evolving adventure.
You recently published a paper titled Pathogen Dilution, Research Partitioning, and Precipitation Generate Productivity Benefits from Plant Diversity. Would you tell us about what this publication is about?
Yeah, I'd love to. So this was, we're reporting the results of the first 6 years of this large experiment where we manipulated the number of plant species in a plot, all prairie plants. The composition of those plants from the same family versus plants from different family, and then the level of precipitation. We removed all the plots are in these shelters, and then we remove all the rain and we put it back on with a sprinkler system. And so the goal is to evaluate what is the value of plant biodiversity. Why are there so many plants? And what is their impact on productivity and functioning of our communities? And so a major thing that came out of that is that interactions with pathogens can play a major role in the productivity of these systems in high diversity communities. Okay.
So I had been up to the field station, and I believe I had seen some of your grid system for these plants. Yeah. If I remember correctly, you had some plants that was a monoculture and then some that were a polyculture. With those two sort of different setups, did you really see big differences between the two?
Yeah, they're exactly right. And the monocultures were consistently low productivity. The total amount of plants in the plots was much less than the productivity of these plots in polyculture. So productivity is something that we can all value. It relates to the agricultural productivity, all of these things that we want, a desirable property of ecosystems, increases dramatically with the diversity of plants, with the number of kinds of species in those plots.
For the pathogens, if it was a negative pathogen, I suppose, would you see more of them in the monosystem versus the polysystem?
Yes. So the pathogen impacts are greatest in these monocultures. Mm-hmm. So the pathogens are specialized, and so they are able to eat some plants but not others. And so in a monoculture where it's all plants that a single group of pathogens can eat, they go from one plant to the next plant. They're always able to consume everything around them, and that suppresses the plant productivity. But when you have lots of different kinds of plants near each other, then there still may be the pathogens in that system, but they are not amplified by their neighbors. So when they disperse to their neighbor, they're not able to thrive. And so then the overall density of the pathogen, the overall impact of the pathogens in the community is much less. So the diverse plant community suppresses the pathogen impacts, and that allows for greater productivity in the system.
Yeah, I mean, that makes a lot of sense. I know you hear about like orange tree blight and how these huge farm systems that are just orange trees, how they'll all be wiped out by one disease showing up. So in a way, it makes sense having more diversity of just things to kind of, like, in a way, kind of make a wall against that to kind of help it spread.
That's exactly right. And so that's a common problem for agriculture, is that it's easiest to plant monocultures of corn and soybean here in Kansas or oranges in California or Florida. But that ends up being just a perfect situation for the pathogens, and which creates very deleterious problems big problems for farmers.
Along with the pathogens, you had also mentioned that precipitation had an impact on growth and loss. So breaking this down for the everyday person, why is it important that we research changes in precipitation and the loss of species diversities in grasslands or other sort of cultures that you're growing?
Yeah, we removed all the rain here in Lawrence, and then we put on one plot, one house, we would put 50% of the rain back on, and that sort of replicates the environment of the middle of Kansas, where it's drier. And then we take, in the other house, we put 150%. So that replicates the more moist environment of eastern prairies in Illinois and further east. And so what we find is that the eastern prairies have a lot more kinds of plants. They have a more diverse prairie community, really at a local scale, than in the central Kansas. And what we find in our experiment is that the productivity benefits of diversity increases with precipitation. And a major reason for that is that pathogens like it wet. So in these monocultures and with a lot of water, the pathogens really build up to high densities, and that suppresses the yield. So here we are in Lawrence, and we're putting a lot of more water on the plots, and we're getting less productivity. But in the polycultures, we get the response that we expect. When we put a lot of water on the plots, we get more plants. And that's because they're able to control the pathogen impacts with the diversity of the communities. And so that illustrates that in areas with higher moisture, then it's even more important. Plant diversity is functionally more consequential for productivity, for ecosystem function.
What kind of methods did you utilize to find your results, and then where did your research take place?
Yeah, so we were working at the KU field station, and a lot of the maintenance of the plot composition is weeding. And so we employed a lot of KU undergraduates over the summer who learned taxonomy, plant taxonomy. They had to learn to identify all the plants, and then it was a lot of work weeding during the summer. But they were involved in a lot of different dimensions of the research, and a lot of those students went on to grad school in the sciences. But we also did sequencing. So in order to determine the composition of the microbial community and the pathogens and other microbes, we would take soil samples and we would extract the DNA from that samples, and we would identify all of the species present, all the microbial species present by amplifying and sequencing the DNA. And that involves a lot of bioinformatics, a lot of technical challenges in interpreting that. But so it's those two kinds of things. One is just like gardening, and the other one is lab work. And putting those two things together is what allowed us to get the inference that these pathogens are the major drivers of the productivity increase with diversity.
Just to reiterate, during this study, what kind of findings did you have? And then what will those findings mean for further research?
The importance of plant species diversity for pathogen control in productivity gains, it has important consequences. We're still trying to understand how those consequences cascade through the ecosystem. So we do see greater carbon accumulation in the soil, so greater nutrient in the soil with higher diversity of plants. So that is one of the longer-term benefits in trying to work out exactly the mechanism driving carbon accumulation in soil is a major avenue for future research. If we better understood how we sequester carbon in the soil in these grasslands, because grasslands hold lots and lots of carbon in the soil. That's why the soils are so black. If we better understood that process, we would better be able to manage our communities to remove CO2 from the atmosphere and help balance the climate problem that we're all facing right now. But the other more practical dimension of this is just directly for agricultural production. Farmers are planting in monoculture. If it was possible to include more kinds of crops in a cropping system, what would be the benefits, and how would that benefit of crop diversity balance with the effort to breed resistance in individual crops? The value of crop diversity is that you can get resistance to many pathogens at once. While breeding for resistance, you're targeting one pathogen at a time. It's a slow process, and the pathogen evolves and overcomes that resistance in continually. So it's possible that we could get greater productivity by incorporating those two things together, and we're studying that now with a grant funded by the U.S. Department of Agriculture.
Is there anything we haven't talked about yet that you think would be good to sort of touch on?
I'll just add that going back to how the plants are interacting with lots of microbes simultaneously, one other avenue that we're exploring now is how interactions with the beneficial microbes might alter how the plants defend themselves against the pathogens. And we finding really complicated interactions, which we're in the middle of working out, and that's something that I would enjoy talking with you in maybe in the future.
Yeah, no, I'd love to hear about it. Thank you so much for joining us, Dr. Bever. I really appreciate you taking the time and stopping by, and for offering to come back again.
Thank you so much. I really appreciate that you're taking the time to talk to me.
Thank you for listening to Interview with the Biologist. You can check out the show notes for more information about Dr. Bever's work, as well as a link to the research paper discussed in this episode. A full transcript of this episode will be available at biology.ku.edu.